This webinar will cover mutation detection and CNV analysis for Illumina sequencing data using HaloPlex Target Enrichment Panels. I'm John McGuigan, and I'm a biologist at SoftGenetics. HaloPlex Target Enrichment Panels have four main steps. First, the sampled DNA is fragmented using restriction enzymes and then denatured. Second, the probe library is added and hybridized to the targeted fragments. Third, the targeted fragments are purified and ligated to form closed circular DNA molecules. Finally, the fragments are amplified using PCR. For a more detailed overview, you can check out the app note on our website, Mutation Detection and CNV Analysis for Illumina sequencing data from HaloPlex Target Enrichment Panels using NextGENE Software. The workflow for these projects is relatively straightforward. First, we perform format conversion, where we convert from a FASTQ file to FASTA files and do some quality filtering and trimming at the same time. Next, we align the data to the whole human genome reference. And finally, we examine the results in the viewer, where we can review, edit, and export many different types of reports. And some of the main reports in this type of analysis include the mutation report, the CNV report, and the coverage curve report, which is very good for quality control. An important consideration for HaloPlex data is that partial adapter sequence exists on the 3' end. This is very easy to handle in NextGENE. During format conversion, all you have to do is select an option and load a text file. And the text file is structured like this. There's 4 columns. The first column is the adapter name. The actual name that you use doesn't matter. The second column is the sequence that we want to trim on the 5' end, which in this case there's no sequence, so the column is just a dash. The third column is the sequence that we want to trim on the 3' end. And in this case, there are 2 different sequences, one for the forward reads and one for the reverse reads, but we do all the trimming in one step. And finally, the last column has a code to indicate the adapter trimming mode. In this case, we use P for partial adapter trimming because the full adapter won't necessarily be in the sequence. The other 2 modes available are E for exact matching and L for loose matching. When NextGENE first opens, the project wizard is shown. Here you'll just select your instrument type, your application, and we're doing SNP indel discovery. And you want to set the number of cores that you're using. I usually like to use 1 less than the total number of cores so that the computer isn't completely busy during processing. So the first step is format conversion, and here we'll load our raw data. You can see that the program automatically selects that this is FASTQ data, and we have the correct instrument selected. You want to set your output location, and then you want to make sure you have the correct format conversion settings. The default settings are fairly close to what we need. We just have to select that this is paired in data and select to trim by sequences in file in order to remove that adapter sequence. And then we'll just click OK. After a few minutes, depending on the size of the data, you'll get a message saying that the format conversion is complete. So then we go back to the project wizard. We load our converted data. You can see that the format conversion produces a converted file and a removed file for each input file. We then select our preloaded whole human genome reference and set the output location for our project. Click Next. And now we set up our alignment settings. So a quick way of adjusting a few of these settings is to use the Inspect Input Files button, and this will take a look at the length of the reads in the input data and automatically adjust some of these settings for the actual alignment. The alignment has three main steps. The first is when NextGENE tries to match the entire read perfectly. The second is when NextGENE tries to match the entire read with a certain number of mismatches allowed. But in this case, we skip the second step because the reads are long enough that it's faster to just use seeded alignment, which is the third step. So in this case, NextGENE suggested that we create 40 base pair seeds every 13 bases. So there's plenty of overlap between consecutive seeds. We can optionally select to detect large indels. This will add some extra processing time, but it can help detect indels, especially near the ends of reads. By default, NextGENE is able to detect insertions up to 1/5 the read length and deletions up to 1/3 the read length, so this option isn't always necessary. For HaloPlex data, we do not want to hide unmatched ends because the way the reads end up in the alignment, since all the reads start or stop at roughly the same position, if there's a mutation near the end of the amplicon, there's a chance that it could be hidden using this option. The mutation filter settings are fairly straightforward, so we can adjust the minimum percentage of reads at a certain position that have to have the mutant, the variant, in order to call it as a mutation in the mutation report, a minimum level of coverage, and a minimum SNP allele count, which is the same thing as multiplying the percentage by the total coverage. This is how many times the variant has to be present. So an easy way to filter is usually just to increase the SNP allele number. So in this case, I've just tried setting it to 10. So this means if we want to detect a 10% mutation, we would have to have 100 coverage. If we want to detect a 50% mutation, we would only need 20 coverage. So using this setting allows for a little bit more flexibility in the total level of coverage. The balance ratios filter is optional. It's not recommended for HaloPlex data because in some positions the reads aren't necessarily expected to be balanced equally in the forward and reverse direction. And the final setting we want to adjust is that we're loading the paired reads data so that we'll keep track of all of the paired end information. On the next page, we can set up the post-processing. So this allows us to automatically generate multiple different reports. And here you just load your settings files for those reports. So what you can do is have NextGENE automatically generate multiple different mutation reports using different settings. For example, you might prefer to have one mutation report that lists the SNPs and one mutation report that lists any indels, and maybe use different filtering settings for each different report. In this case, I'm not going to set up any of these reports, but I will show those same reports in the actual viewer. So then we just click Finish, and we have the option of running this project right away, or if you have a couple projects, you can go back and create more projects and just change the sample data and output location. And that's easy if you have a handful of projects. But if you have a larger number of projects, it may be easier to use the Autorun tool. So now I'll just click Run NextGENE, and the processing will start. The alignment will be finished relatively quickly. Again, it depends on the size of the sample data. So this project took about 20 minutes to run, and after the project is done running, it'll open up in the NextGENE viewer. So here we can click and drag to zoom in and out. I can skip forward between mutation calls. And so the first thing to go over are all of the mutation report settings. In this case, I had previously set up the DB NSFP and Cosmic databases, so I can check to display the Cosmic data, and on the DB NSFP tab, I can display the classifications for the functional prediction scores from the DB NSFP database. For filtering, since this is a targeted sequencing project, it's very helpful to filter using inclusion for the regions that we were targeting. In this case, I already have a bed file created that specifies the regions. So now I can just hit OK, and any mutations that are not in the target regions will be removed from the mutation report. So you can see that most of the variants in the report are purple, indicating that they are in the DB SNP database. And in total here, there are about 61 variants. The coverage curve report is very useful for quality control. So I'll open the settings, and here we'll load that same bed file that we used in the mutation report, and we'll set a minimum coverage setting. And since I used 10x SNP allele coverage, I think a coverage cutoff of about 20 should be relatively useful. So now we can see regions that do not have at least 20x coverage, but were included in our bed file specifying the regions of interest. And all of these regions are described down here. And even more useful is the coverage curve summary report. And this will show you a graph of the coverage. So if we zoom in, we can see that approximately 95% of the targeted regions of interest had at least 100x coverage. In the table down here, we can see that over 99% of the reads were able to align, and approximately 92% of the reads were on target, meaning at least half of the read was in the region of interest according to this bed file. The average coverage was very high, and almost 99% of the regions had at least 20x coverage. The last topic for this webinar is the CNV tool. And the CNV tool can be opened from the Tools menu in the NextGENE viewer. The CNV tool always uses a comparison between two samples. One sample is considered the test sample, and the other is considered the control. In this case, I know that the E project has a known CNV in the KCNH2 gene, and the F sample does not. There are three different options for comparison. The comparison regions could be specified using the built-in annotation, so by coding sequence, mRNA, by continuous mRNA, meaning there's one record per gene, but it doesn't include the introns. Continuous CDS, which is the same, only it's based on the coding sequences instead of the exons. The second option is to set an incremental length as small as 100 base pairs. And the most relevant option in this case is to input a bed file, which is the same bed file used earlier. And this way we can compare the coverage in each of the targeted regions. So currently the only coverage measurement for the CNV tool is using our own SNP-based normalization with median coverage. This will perform a global normalization based on the median coverage of the regions and the projects. Lots of different annotation descriptions can be shown in the report, along with the original coverage, the normalized coverage, the log2 ratio, and the z-score. The way the normalization works is that several different positions are checked for each region, and one position is checked to be representative for that region, so you can see what position was selected and what the allele frequency was for each of those positions. We preferentially select a region that has a mutation at that position in both projects, so that way we can compare the change in allele ratio in order to best normalize the data. There's some filtering built into the CNV report. You can set a log2 ratio filter so that anything more extreme than this range will show up in the report. So in this case, I'm going to set a range from -0.7 to +0.7. So the way the log2 ratio works is -1 would be an ideal deletion, meaning there's half the number of reads in the test sample as there are in the control sample. A score of 1 would be a 1 to 2 duplication. And in general, deletions are easier to detect because you're going from 2 to 1, while duplications are a little bit more difficult because sometimes you might be going from a number of repeats to just a slightly higher number of repeats. For example, duplication that amplifies a certain sequence from 4 copies to 5 copies would only increase the number of reads by 20%. So that can obviously make detection difficult since there is some variance in the number of reads covering each region even after normalization. The z-score indicates the confidence that this change is real, and it's based on the amount of coverage, the maximum score being 20. We also filter out regions where both projects are low in coverage, so we require that at least one project has a minimum level of coverage. And in this case, I'll go ahead and set it to 100 because we have very high coverage in these projects. And then you just hit OK. And in this case, the CNV report took about a minute to open. So here you can see the CNV report once it's done opening. This description column comes from the bed file with the fourth optional name column in the bed file. We can see that we did actually find the expected deletion in the KC and H2 gene. And we can see that the log2 ratio was very close to what we would expect for a real deletion. And you can see that there really wasn't much normalization needed. The amount of coverage in the control sample was lowered only very slightly. So that concludes the webinar. You can visit us on our website at softgenetics.com. There are several different webinars available for viewing in the Analysis Corner link on the website. And there are many different application notes where you can find more specifics about particular topics. Thank you.